Okay. I think we are ready to start tonight. So thank you for being here. This first waiting for Pint 23. Tonight, tonight we will have one of our. Yeah.
Are you starting or practicing or starting?
This is always like this, but it's kind of normal.
Yeah.
No, it's not frozen. It's
Somebody's working, somebody's not.
exactly.
Imagine.
But wait. I'm sorry.
So, ladies and gentlemen, I have a question. How many of you are Norwegians? Not that much. Because usually we have the participation of the international people, not so many Norwegians.
Yes. Here we are. I'm sorry.
This is the question that we're asking ourselves.
Yeah, I'm sorry. So again, welcome to Pint of Science. This first waiting for Pint 23. I would start this night very quickly introducing what Pint of Science is. Pint of Science is an international organization working on science communication to the wide public, to the general public. And what we try to do is to bring science to everyone, and in places where we can relax, we can enjoy a nice pint of beer, but at the same time also listen to researcher actively working in our science environment, in Oslo in this case, and update us with the newest, let's say, the newest results, let's say, of their research activities. And this is very important, of course, for science available to everyone. Pint of Science started in 2012 in UK, and then it very quickly spread all around the world. This map is not updated. It's updated to, it still is the official one, but it's updated to 2019. So now we have 26 countries in Pint of Science. In Norway, we have 5 teams actively working every year, creating the annual festival. Every year, Pint of Science organizes a 3-day festival in May. This year, in 23, it will be from May 22nd to May 24th. And of course, we hope you will come, and you will also enjoy these 3 days of science. And everything we do, it wouldn't be possible. Everything you will see today, tonight, and also during the festival, and also during other activities that we always try to do during the year. So nothing will be possible without the team. This slide is not updated because, for example, you cannot see Daniel. That is one of our new volunteer. And we have also Bushali online that's helping again with the IT. But everyone is working hard to make this thing possible, giving her free time to this initiative. Everything is is created by volunteers here. So I want to thank all of them, everyone who is working with me in this initiative, and of course our speakers, because we are volunteers, but also our speakers are doing a lot, and we know that it's particularly difficult to create a presentation like this for science communication and not for science dissemination. So we really appreciate your efforts to be here tonight, and we really want to thank you a lot for this. The last thank goes to our sponsors. As I said, we are all volunteers, so all the money we have, everything we can do, comes from our sponsors. Simula is supporting Pint of Science since Pint of Science Norway since the beginning in 2020, and Brygg is also supporting now because this space today is given for free. So thank you very much. And three, of course, important things. First of all, if anyone wants to join the team, everyone is always welcome. There's always work to do. For the next festival, we opened a call for speakers. So if anyone wants to join the festival as a speaker, please check the website, check our social media, and you will find all the information there. And if you want to support us, we are very happy to accept donations on Lips. And now I think I can leave the floor to Sean.
Oh, quiz now.
Yeah.
But they won't know any of the answers yet.
Well,
that's
I know we do the quiz at the end. Oh wait, I come around that way.
It's like the quiz is answering questions why we need to listen to the speakers.
Okay, so if you go to menti.com on your phones and enter this code, then you should be able to join. Yeah, the code is on there as well.
Oh.
Yeah,
Hello?
yeah, showing the
It's showing the answers.
sorry, I'm sorry. Well,
The first
one.
The first one is probably one of the only answerable ones, I think.
Yeah.
So we've seen the presentation. Oh, someone got volcano.
What was the
The code is at the top there.
It is.
87817626. And like the snowman emoji as well. Okay, let's see. Thank you. Some here? Okay.
Oh.
Emojis joining, joining. How do I? When I want to go forward on the slides, what do I press?
Usually the space bar.
This one, okay. Okay. Is everyone in? Oh, no, it's still new ones joining. I'm disappearing. Okay. Oh no, why are more people disappearing now? Okay. Is everyone in? Speak now or forever hold your peace. Okay, a moment. Yeah. Oh, Giant Donut has joined us. Yay. Okay, say when you're ready. Where is it? It's in the Menti meeting. I feel like we lost some again. Oh, and in again. Show the presentation. Right?
Yeah.
Okay, yeah. I mean, as long as that, if I press space, yeah, there we go. Okay, I hope everyone's in then. First question, this one you already had the sneak peek answer to. The faster you answer, the more points you get, so fastest speaker first.
The streaming. Yeah.
This one.
Nice, very quick. Yay! Everyone got it right. Okay. Then we go straight to the next one.
Wait.
Oh, no. It's here.
It's there. It's there.
It was working before. Yeah? No? Yeah? No? But will it work now?
So you just show
It is,
Yay.
I don't know why it lost.
Okay, question 2.
show some answer.
Yeah, the stream.
Who wrote
Okay,
The Fable
yes.
of the Fox and the Grape? See, before we've seen any of the presentations, this probably isn't going to make much sense. But have a go anyway.
Was it working before?
Yeah, for you.
Okay.
Yeah.
It
was
Fox and the Grapes.
Okay, well, for now.
Oh, okay, wow. Most people are on the ball here. Okay. I feel like we have a specialist audience. Very specific. Okay, this is how we're doing. Nice. There's everything to play for. All right. Question 3. Answer fast. Autophagy relies on the action of which organelle in mammals?
Sean. S-I.
Let's see if we get more of a spread on these answers.
But there is, you see here, I put all the banners.
Oh, okay. A bit of a mixed bag there.
There are banners.
No, but
Yeah.
it should have been lysosomes, and I guess we will
Yeah.
find out when we listen to our presentation.
Night. Yes, I
Okay.
forgot, I'm sorry. So there
Next
is Sean.
question.
Yes.
How many neurons, brackets nerve cells, are in the human brain? 8.6 billion, 86 billion, 860 billion. Either way, it's a lot, I think. Anything in the billions is just uncomprehendable anyway, so. 86 billion! Most people knew that. Oh my god, okay. I was pretty shocked when I put that in actually. As a question. All right, things turning around at the top. Okay, question 5. Halfway there, or nearly halfway there. Oh, no, we've gone back again. Oh, wait, what happened to question 5?
You will not go back, I think, you know. Oh, yeah, you will. Enter.
Okay, there we go. Which embryo is this? What is it? A fish, a chicken, or a human? Who knew they could be so difficult to tell apart? Human! Yes! Not many of you thrown off by the gills. That's maybe worrying, actually. Okay, question 6. Which household appliance is like a black box AI? Ooh. Toaster, dishwasher, vacuum cleaner. Yeah, dishwasher. Vacuum. when you know was popular.
Maybe the automatic robot.
Oh, it's turning around at the top. Audio breaker. Who's audio breaker? Passionate. Okay, question 7. Yeah, this I can't even read this question out because it makes no sense. How many rainbows does it take to jump from Hawaii to 17? This is, yeah, I'm confused. Oh my God! Yay! But apparently it's also 2 rainbows. I don't know. There's something else we need to find out in the talk, I think, isn't it? Audio breaker's still killing it at the top, I think. Uncatchable. Question 8. Which of the following best describes autophagy? Cell breaking down its own components, cell digesting a foreign bacteria, cell going through programmed cell death.
So.
Oh, quite a spread on that one. No, apparently it's a cell breaking down its own components. All right. Nothing can throw audio breaker off the top spot at this point. All right. Question 9. 2 more questions to try and make you break for the top spot. Throw audio breaker down. How many artificial neurons were there in the 2012 AlexNet image classification network? Fastest finger first. All right, time's up. 500,000. There we go. Most people knew that as well. Any changes at the top? No. Through a few people falling down the leagues. Right, last chance. Let's see if Helen can try and beat audio breaker. Question 10. What? Okay, well. Yeah, this would have made more sense at the end. But have a go. Predict the future.
But it's kind of famous. It's kind of famous.
Yeah.
So,
What are you going to see?
Amazing.
Some people are psychic.
Final standings. And I think it's audio breaker. Yes.
Well,
Outstanding performance. Is that someone in the room?
yeah. And I made 3 other questions, so let number 2 win.
There we go.
Go to number 2.
Ta-da.
Helena.
Helena. Okay, who's Helena?
Yeah.
Okay, number 2.
you also have prizes.
Yeah. All right. Now, hopefully, that served as a good teaser for the actual presentation we'll now see.
Thank you.
Got your mic back.
Okay. All right. Welcome everyone to Med Night. So my name is Mar, and I'll be introducing the speakers for the night. So we have 3 sessions with a break in between. And we're going to start out with Amani Outa. She's a pharmacist, and she holds a PhD in biomedical sciences from the American University of Beirut. And during her PhD, she developed and validated the first fruit fly model of human chronic myeloid recipient of the National Council for Scientific Research of the Lebanese Republic, doctoral studies, and the Lucca European School of Hematology Scholarship. And currently, she is a Scientia postdoctoral fellow at the University of Oslo. So if we get her presentation up and running, and you can have a microphone. Okay, so give it up for Amani.
Yes, so hello everyone. It's my pleasure today to be presenting my talk. Thank you for the nice introduction, and thanks for Pint of Science for giving me the opportunity tonight. So the talk is entitled Autophagy-ON Autophagy-OFF, a Time Drive. You have probably once in your lifetime hopped on a hop-off bus and explored a city. But tonight we're going to explore a different topic, which is autophagy. So the ride is to explore autophagy, and for that, I would like to introduce you to Mark. Mark has a very important task, or he is thinking about an important task to do, whether or not to clean this messy room. And to the right, I would like to introduce you to one of your body cells, also thinking about whether or not to clean itself, biologically known as autophagy or cellular housekeeping. So Mark has to daily take care of his room, clean it, get rid of the waste straight off the room. That's to keep it going on well, or to keep what we know as homeostasis. And also your cell, the cells in your body need to do the exactly same job in order to maintain cellular homeostasis. So the same aim for Mark and the cell. But for the cell, and also hopefully for most of us in this world, it's not only about dumping the waste and or trash bins. It's also about recycling it and making something useful out of it. And that's what cells do most of the time. So here you are seeing the whole process of autophagy as it goes, or the main events. And to make an analogy, the cargo here is the waste material, or the material to be recycled or degraded. We call it cargo. And then these are the recycling bags, let's say, which we call autophagosons biologically. And they start forming to enwrap this cargo, and they form this very nice and fully mature autophagoson. Then a very important event takes place, which is the fusion of this recycling bag with a helper. This little guy here, the lysosome, that's important for autophagy. This lysosome brings with it tools to degrade that cargo or recycle it and make use out of it. So after the fusion events and the whole thing going on inside, then there is the material that is useful for the cell, or the recycled material is given back to the cell to make use of it. And in this way, the cell would have survived something, or at least made sure that the processes are going okay. So basal autophagy, as we call it, which might be going on in your body right now as you're listening to me, getting rid of unuseful material, getting rid of toxic substances so that they don't build up, so that diseases do not arise, is a very important thing that should happen on a daily basis, and it's important for the cells to function normally, just as you straighten up your room every day so that the house is working okay. So both the cells and Mark are happy with basal autophagy going on. But what if stress or chaos occurred, which for Mark could be a wild party at his home, and there is more waste to take care of, and for the cells could be low energy levels, low oxygen, or low nutrients around. Then the cell is shocked and has to respond, and the response is usually increasing the level of this basal autophagy to match the stress, or we call it activated autophagy, or higher levels. And that's because if the cell doesn't do this and does not respond, diseases could arise or the cell could die. So if we think about Mark again, if you have a party and you want to clean more, we could either increase the number of waste bags you are using, just as marked here, or you could bring a very big bag and put everything inside. And like this, you have gotten rid of the waste. For the cell, it's exactly the same. It could increase the number of these recycling bags or autophagy-ons, or make huge ones to accommodate for the bigger cargo to get rid of. So upgrading the whole process. But it's very important that while Mark is cleaning his room, he doesn't go beyond removing the waste and removes the furniture or what is needed for the room to be functional. Then the room, of course, will no longer be okay. And this is not what we want, sorry. So the cell, if it starts autophagy and it upgrades the whole process to get rid of the excessive stress, it has to stop at a certain point, or otherwise this will lead to a disease, or the cell could die, or the room will become non-functional again. So as it activates autophagy, it has to stop at a certain point. So that's what we mean by with the time drive. But also at the same time, on the other side, if too little cleaning is occurring every once in a while, then there will be buildup of toxic substances, leading also to diseases, but also for Mark would be a smelly room. So the important thing is that autophagy has to be tightly regulated. The response to stress has to be matching to the stimulus, if you want. So, if we go to the analogy of the hop-on, hop-off bus, then this means that the genes in our body that hop on the bus for the autophagic ride, they should, so the ride can start, they should hop off at the right time so the ride could stop. And if you look through literature, we know a lot about how autophagy starts and which genes and pathways are involved in getting the ride to move, but we know little, surprisingly, about how does it stop, which genes are involved, or mechanisms or signaling pathways. So the main question is, which genes could be involved in stopping the process, which is what we're interested in. And before we know which genes, it's how we're going to know about these genes. And for this, I'm here in Norway studying this. I use fruit fly cells, and a microscope to screen and look for genes in these cells that could play a role in stopping autophagy. All of this done while silencing the gene of interest. And the gene of interest is silenced individually. So, in other words, I challenge the cells, I starve them, which means I remove the nutrients from their environment. And this is a stress for the cell. So the expected outcome is a response, which is autophagy or activation of autophagy. So autophagy is on. But then after a while, I refeed the cell, I give it nutrients back, and the expected outcome is that it will be satisfied and autophagy should stop, or autophagy is off. But this is done while individual genes of interest that we would like to study are silenced. What's the importance or significance of that is that you will know then the role of the gene, whether it plays a role in stopping autophagy or not, or how the ride would be. So back to the bus, now the genes cannot hop on normally or hop off at the right time, and then we would like to know, how will the ride be? Will it be faster to stop, or would it stop earlier? It depends, of course, on the contribution of that gene. So how do we study that? If we zoom into the cells, to the autophagic process, then we decided to follow this guy here, and it's known as ATG8 in cells. Let's call it the white guy here. And then we gave it two colors, a flag with two colors, red and green. And why do we follow ATG8 or this guy here? Because it's usually in close proximity to these recycling bags. So if we follow it, we can follow the whole process along. And as long as these bags or recycling bags have not fused with the lysosome, then the color is yellow because it's a mix between red and green, and it will fluoresce as yellow under the microscope. That would help us to track this guy. So, but if the autophagosome or the recycling bag fuses with the lysosome, then there will be acidity inside due to these tools or enzymes to degrade the material, and then the green color disappears, and we're left with red color. So what this means is that as long as we have yellow recycling bags, we are having either basal autophagy or not so much high level. And when we have red bags, this means the process has moved beyond and the process is ongoing. So yellow bags versus red bags, and this will help us track the whole process. How do you see this in real life? You are seeing here a microscopy image of around 30 cells of Drosophila cells. Each rounded object is a cell, and inside it there are spots. These spots here are yellow. So if you guess right, then it's a fed state. The cells here are satisfied. They're not engaging too much into these fusion events because they don't have to. So it's a fed condition. And if you look at this cell, for example, you would see these are the spots, and this is the whole cell, the yellow bags. Then, when I challenge these cells and I remove the media, the nutrition from their surroundings, then it's a stress for them. And the response is autophagy, which means more of the red recycling bags or red spots as we see them under the microscope. Again, a cell with the red spots inside. So which means that autophagy is ongoing, and the number and the size indicates that the level is high. Yes, this is how it looks. And then if we quantify this as percentage of cells in autophagy versus time in hours, if you follow the yellow line, it's a line of a condition of cells that are satisfied, nutrients around, no need to activate autophagy, then the percentage of cells in autophagy is low. However, when you challenge the cells with starvation or removal of nutrients, they shoot up with the number of cells in autophagy, as you see, much higher. Then, if you keep them, so autophagy is on. After that, if you keep them starving in the red condition, then the percentage of cells in autophagy is more or less the same. Autophagy is still ongoing for a long time. But if you refeed, you can find that after around 2 hours, there is an exit in autophagy in some cells, which stops the process. So the percentage of cells in autophagy decreases, stays more or less the same. While the fed state all the time is just low with cells needing to activate the process. So back to the analogy of the bus, this autophagy off, of course, in this part, this is how the ride would look like. Not working? No. Okay, it should just go down. So this is how the ride would look like, the normal ride, when genes could hop on and hop off at the right moment. But when we silence the genes and then we do the exact same thing, you can expect that the ride would stop 1 hour before, 4 hours after, slower or faster. It depends on how much the gene is important for the process. And with this cartoon, I would like to end my talk. Now we are in the phase of analysis of several genes involved in the process. I hope next time I'll have some interesting hits to discuss with you. But I hope that now you know why and how our cells eat themselves and why it's important to study autophagy for treating diseases. This work is being done at the Institute for Cancer Research at the Radium Hospital, and I'm a Scientia Fellow at the University of Oslo. It is done in the answering group with Helena Knabelsrud as my supervisor, who is here now watching. And I would like to thank the funding sources and the collaborators and the team and you for listening. Thank you.
Thank you very much, Amani. That was a very interesting talk. So are there any questions in the audience? Yeah, so I will actually bring the microphone to you. So the people streaming here.
Okay. On your graph that showed when the cells were refed, then the autophagy goes down again. But why doesn't it go back down to the original fed level? It stays even quite high once they're fed again.
Yeah, we experience this hybrid population of cells. Some cells do really activate the process to a higher level, and some cells do exit in a better way. Some cells do keep the process going. It's actually an interesting question which could lead to other sub-questions, like why do different cells within the same condition respond differently? So yes, we experience that.
Anyone else?
I know you're studying autophagy kind of on a basic level, but what do you think about like the recent trend of like us humans like going for fasting and like extended periods of no food, like to like increase the autophagy?
I think there has been an association between fasting and activating autophagy levels. However, it needs lots of research to determine the optimal time and how much, at what point you start benefiting from it and what times you should stop and refeed and the cycles. Interesting topic is starvation and refeeding, and it helps you recharge, and then we go into another cycle with with more benefits. But of course, this is like a bit far away from my own knowledge, and we would need more reading on that.
Other questions? Yes.
Thank you so much. It reminded me my biology classes actually. I understand it's a basic research, but actually how it can be used for like the real life for the person on the streets.
Yeah. Interesting. So the ultimate aim is when we unravel novel genes that could play a role in stopping autophagy, for example, then when we want to use autophagy as a therapeutic target for any disease, because it's associated with several diseases, then we could know how to manipulate or have better control over the process to stop it when we need to, or elongate it when we need to, and then this is the flexibility that we are getting, hopefully, as targets for drugs.
Oh no, I'm going to debate.
No, I'm not going to debate. I'm just curious if you thought if there's like maybe in your field some thoughts about cancers to get autophagy to eat a cancer. Is that something that can be done at some point?
Yeah, so in cancer, it's a bit like a double-edged sword. Autophagy plays a role, like whether it's a precancerous or it's an established tumor. It's still a huge debate, like in the field, how to really tackle autophagy in cancer, and I think it's highly dependent on the type of cancer, so it cannot be generalized. But yeah.
Okay, one more question. What will be your next steps then?
Next step?
Yeah.
I hope now I'm in the middle of trying to get the best analysis of the screen for many genes. So I hope that I will get the way through to find some hits and then study these genes on a more detailed level.
Okay. Any more questions? Okay. Thank you.
Thank you.
All right. So now we will have a 10 minutes break. So we'll be back on 5 to
10:25.
5 to half on.
English, Norwegian.
Is it possible to have a presentation here?
Yeah, so here I try to show you about myself, but it could be any other scientist because that's how we do science. We move around, live in some countries for some years, and then move around, learn stuff, keep exploring. And we stop at some point when either we like the place or when we don't find more opportunities, essentially. So right now I'm here and having two positions, one research and one scientific communication. So essentially trying to make life out of what I'm doing today here. So what do you think? This is my 5th year in Oslo now. Am I still in Oslo because I like it, or? So yeah, could be. So people have day jobs and evening jobs, but I have right now 3, all in the day's work. So of course I'm a scientist. I'm just starting to be a scientific communicator and communication advisor now. And surprisingly, I'm a cricket player for Norway. I play for the national league,
Hey.
and it's quite fun. So I like Oslo. So you saw the title, from egg to embryo and then back. And what do we do when we don't know something? We do a Google search. So if you search for egg, this is what you get in the images. Yeah. No information about embryo here, just the egg. There is one little picture here when the baby is coming out of the eggshell. So I thought, okay, maybe if I go there, I find some information about embryos. I click that. Still no real information about how the egg becomes an embryo, yeah? So not very useful Google search. If I search for embryo now, then now you start seeing all these complex pictures, yeah, of developing embryos of mostly human babies or some other babies. So this is what we are trying to study. And how would you study something like this? We make use of model organisms because, of course, we can't study ourselves, yeah. And here you see some of the most famous model organisms that people use in the labs to study. So starting from a round worm, fly, which Amani already showed you, fish, frog, chicken, mouse. Some people also use monkeys. People do research on bears. So as you saw, Google did not give you any information about egg to embryo, but I just wanted to show you one movie of a fast developing animal, which is zebrafish. The scientific name is Daniel Radio, and then we will see it on YouTube. So YouTube is still useful. So here you saw these are few cells here on top of the yolk, and these are very rapidly dividing. This is real time, yeah, just dividing into now these are like 4,000 cells here already within 4 hours, and then the cells will start moving down, covering the yolk, and then they will extend on one side. And then that extension will lead to formation of tail on one side, which you'll see in a moment. Yeah, this is the extension. And the tail will appear somewhere. Let's see where it appears. Yeah. So this is the head now. Yeah, that's the head forming. And the tail is going to appear this side. And these are two completely different embryos, but just look at the sync. They are growing completely in the same time frame. Yeah, and this is like the backbone of the fish. And that's the tail, here is the eye. And try to see here. You'll soon see the heart pumping, and there's just 24 hours into it. You'll see a pumping movement here. Can you see that? That's the heart pumping, and very soon the blood starts to flow. 30 hours from a single cell to an animal which has all the organs. That fast development can be in some of the animals. We don't see it in humans because it's in utero, it's inside the uterus. We don't see it. Yeah? So let's go back to the presentation then. Right. And then in 3 months' time, these are adult zebrafishes. That's a female, and this is a male. And they are ready to reproduce, go back to the egg. Yeah? What is fascinating is this egg and, of course, the sperm. They combine together to give rise to a whole animal, but retain the ability. So these cells come together, make a whole animal, all the organs that you can think of, but still retain the ability to give rise to the next generation. So they keep their, let's say, if we call it as an egg quality. Yeah, in biology we'd call it as a germline. So egg and sperm together are called as germline or germplasm. They retain that ability. And if that ability is not retained, let's say these two fuse together and just get converted into all other organs and don't keep their own property, then essentially life is lost, right? So this is the fascinating point, that how this germline converts to the soma, which is the body, but still keeps the germline property in the meanwhile to give rise to the next generation. So how does it happen in some of the simple organisms, like the worm, which is called, the scientific name here is C. elegans, and this is the fly, Drosophila. The red is the germline, or that part which will go on to become ovary or testis. Yeah. So in the C. elegans, one cell embryo, it's spread all throughout. But then when the first division takes place from 1 to 2 cells, all the red dots will go to one side. And they keep doing that. And all that, like kind of after you have around 32 cells or something, all that which will make the next germline or ovary or testis is there in one cell. And it stays like that. And it kind of, it's outside there, but then it moves inside where the gonads would form, where the ovary or testis would form. And then in the adult worm, you have this organ which will produce the next generation. In Drosophila, it's even dramatic that when the egg is not even fertilized, the sperm is still outside, that part of the egg which will go on to make the ovary or the testis is already separated. It's not mixed with the rest of the, it's still one single cell, it's not divided. But that part of the cell which will make ovary or testis is in one side already, separate from the rest which will form the rest of the body. Yeah. If we look at zebrafish, something even more dramatic. So again, like Drosophila, it is like an egg. That part of the egg which will go on to form ovary or oocyte is separated. But when the cells on top of the yolk start to divide, this germplasm divides for 2 times, and then it's there in 4 different corners. And these cells will keep on dividing, the cells will become smaller, and there'll be 4 clusters of cells which will eventually become the gonads, or the ovary and the testis, yeah? And so this is around 6 hours after the egg has started to divide. Between the 6 hours and 24 hours, all these 4 clusters of cells will migrate inside the embryo, find their place, and go where the ovary and testis will form. So all these cells are guided to the one place, right? How it happens in mammals? It's not known very properly because it's difficult to study. These embryos are inside the uterus of the mother. It's difficult to study. The first time when we can see germ cells, or those cells which are going to give rise to gonads, it's around embryonic day 7 in the 3rd week. And then these cells will eventually, same like zebrafish, migrate to the place where the gonads will form. But how these cells are formed is not properly known. All the current experiments suggest the cells on top of it, they signal, provide some signal, and make these germ cells. So it is thought that in the mammals, it's not like other organisms. The rest of the embryo guides which cells will become ovary or testis. But more recent work, because now we can take individual cells and we can do sequencing on the DNA and the RNA on single cells, those experiments now suggest that they actually come from some of the earlier cells. So maybe it is decided earlier than where we can detect it. But this is still something not properly understood. So what is interesting, I showed you different types of how these germ cells are formed and how. But if you look at the molecular details, don't go into too much of details, but in like Drosophila or the fly or the worm or the mouse, you'll see some of the proteins which are required for this process, like nanos. It's there everywhere. And also some of the marks on the DNA, which is required for formation of the germline, is also the same in all animals. So it starts differently, but it ends in the same way. Now coming back to the egg, it's a very special cell, right? This is what continues life. What is interesting about the egg cell is, like in us, when a female child is born with a certain number of eggs already. So even before the birth, a female baby has a certain number of eggs. And these eggs do not go further in development until puberty. So the eggs are there before birth, they wait until puberty to kind of start developing further, but they don't mature up until fertilization, right? So some of these eggs will become mature. Let's say I have a baby when I'm 40 years old, then this egg has survived for 40 years with all the information that is required to make a baby. So that's why I call this as this program that gives rise to animal from an egg. I think it's the mother of all the programs that we can ever think of. Yeah? What happens when the program goes wrong? This is a X-ray of the ovary, where you see teeth-like structures inside the ovary. Yeah, these are teeth-like structures. This is the CT scan of the same patient. These are the tooths inside the ovary. So essentially, the eggs, the program was not working properly. The egg started to make embryo already and started to make all of the organs. And then in this case, it was tooth. In other case, it can be any other thing. Yeah, it's a bit gross. That's why I showed you the just the scans. There is a real image which I'll pass on very quickly. Okay, all that ugly stuff is gone. Now you'll see these pretty worms. So we are not studying that in humans, of course. We are using this tiny little worm, which is known as Sanohaptitis elegans. It's 1 millimeter long. So you can see them by eye, but of course you can't see all the details. You need a microscope. So it's a hermaphrodite. Hermaphrodite means the same animal has eggs and sperms both. So it doesn't require to mate. Yeah. So this is one of the gonad arms. This is an adult animal already. It's a tubular structure. There are small cells here which then develop into oocytes, which are these bigger cells here. Yeah. And then there's a bag of sperms which was made earlier already. So it first makes sperms, stores it, and then start making eggs. And then now this egg, when it's mature, it will push out through this bag of sperms, gets fertilized, and then you have the uterus here where the embryo is developing. So you'll start seeing like this embryo has 4 cells already, yeah, is developing, and this is the vulva, the opening from which the egg will pop out and then start living freely. Very simple organism. It can have the same problem as human. When the program here goes wrong, so these are the nice eggs, the big cells. When the program goes wrong, it's just a mass of cells here, which is a tumor, essentially. So you see, these are the big cells where we have stained the DNA of the cells. And here you see a lot more cells, and it's a tumor. And it's a special kind of tumor because it just does not have one kind of cells. it has other cells, like the red one is muscle and the green one is neurons. There should be no muscles and neurons inside the ovary, right? Because these are eggs yet to make the next animal. So we studied this using this animal. So what is the program and what is getting misregulated? This is the simplest way to explain all the information that dictates life. We all have DNA. DNA is transcribed into RNA. So it's like still the same language, just another way to write it, RNA. And then it is translated into protein. So now the language is changed. It's translated to another language, and that's proteins. And these are the things which work in your cells, like the ATG8 that Amani said, that's protein which will work. Yeah. This is simple, but in reality, it's very, very complicated. So I want to show you this picture just to say that while this is the path, how the information is decoded, but it's actually very, very complicated. Yeah? Now it starts to work. So at which level the egg has its program stored, and at what level this information is now being regulated. So the special thing about this egg is, as I told you, it's in process for several years, at least in humans, right? 20, 30, 40 years. So when it starts, the DNA, so here the DNA is in blue and it's kind of open. Yeah, it's like a interwoven mesh. It's open. It can make RNA. But during this process in which the egg matures, the DNA is compacted in these kind of chromosomes, which is very important because we need to go from a doublet of chromosomes to singlet of chromosomes because the other singlet will come from sperms. Yeah? So for that process to happen, these are first compacted and then separated out. And in this situation, the DNA cannot make RNA anymore. All this process, while it is happening, RNA cannot be made. So before this process actually starts, a lot of RNA is made and stored in the egg. So egg is full of information which is waiting to be decoded at RNA level. So at here. Yeah? So imagine if you want to stop something, it's easiest to stop before starting. But in the egg, it has already started. And you need to stop until the right time. Otherwise, everything goes wrong. So, as I said, a lot of material is stored. So you will see egg actually grows almost more than 100 times during its development. It's one of the biggest cells we have with all this information which is loaded by the mother inside the egg. So again, now coming to the very end of the talk. So this program is now under this RNA-based regulation. And again, it's a simple picture where DNA is made from RNA inside a vesicle, which is called as nucleus. It is transported out into the main cell compartment where everything else happens. And it can be two simple fates for this RNA. It can either get degraded, so make no protein, or make protein. Yeah, that's all RNA can do. And these things can be, of course, these are the things which are regulated. When the RNA should get degraded, how long it should stay, when it should be converted into a protein. And this is how this germline to soma dichotomy is maintained. But again, this is a very, very simple picture. The RNA is regulated at different steps during this process, and it's, again, super complex. And this is what we are studying, how RNAs are regulated during this process of egg to embryo conversion, and how the quality of this egg can be retained where the rest of the embryo is going to become into different parts of the body which are actually mortal, whereas the germline is immortal, it never dies. And then I want to leave you with this picture. These are twins from an embryo that was frozen 30 years ago. So all the information survived in the freezer for 30 years. So as per the record, this is the longest time something has been done like this. There might be other incidences like this, but it is not recorded. So that's the miracle of life. Thank you for listening.
Thank you very much, Pooja. Any questions? Okay. Thank you for-
We have
More questions?
it's always.
How common is it that something goes wrong in this process? What do I need to know the stats now?
We all are alive here, yeah, very rarely.
Okay.
It's a program which has a backup and a backup and a backup. Right? That's how life has survived for billions of years. If it doesn't have a backup, then it's very difficult.
There's one here.
So you should have, when we have an embryo of a female, it's already contains the egg, right? That it's gonna have for the rest of his life. It's not gonna be more or less. Right?
Yes.
I wonder if this ovaric reserve that a woman has, and then it just diminishes with time. Like, is there like a, are we closer to the, I know that there are some hormones, like some parameters, this anti-neural hormone, for example, to measure like, how much of this reserve a woman has. But it feels to me that we haven't done much progress to kind of understand really how much of the woman is fertile and what determines that. So it feels to me that we are kind of a bit stuck in researching that. There's not much progress.
Right.
Yeah.
So yeah, it is a certain number of eggs that we are born with, and as you may realize, one egg matures every month, right? And then once all the eggs are finished, then it's menopause. Yeah. So all the fertility measures are just to measure the cycle, so of the maturation of the egg over this month. Yeah. So they're not direct measurement of how many eggs are still there. It's how good the cycle is running. Yeah. And of course, aging and fertility is a topic of research which people are working on. But we are not. This is something very, very natural. We will not be able to change it.
Yeah.
Yeah, it's very biological and very natural. I don't think, in my opinion, I don't think we can make a 40-year-old woman as fertile as a 25-year-old. No. Yes.
Yeah, this is no. I don't really understand how can you define when you're fertile or not? If people may have eggs and produce them regularly, what can make someone more fertile than others?
I'm actually not sure about this. Yeah, it's something beyond my knowledge.
Yeah, because I can see for men, it's like if you don't have enough sperm, if the sperm is really bad, then you're not fertile, right? But for women, if you have an egg, then why it doesn't sperm?
Yeah, I really don't know.
Okay.
Yeah, I don't know. Yeah.
Okay.
Okay.
Thank you very much. Okay, we're getting ready for the final session. many other things. So please welcome Gabriel and Paul.
We brought along our moderator.
Yeah. Welcome, I'm Pedro. I'm a colleague of Gabriel, and I will give you a little bit of a context about this discussion, which is about explainable EI in medicine. And what does this mean? AI, as you may know, it's very used nowadays in many fields, and it's very timely, this discussion, because can we use AI in, for example, diagnosis or in medicine in general, even if this AI results is not explainable? Meaning that a lot of AI works as a black box. So you just put some data in and you get some output, maybe some classification, and this can help us with the diagnosis, but we don't understand what's happening inside the black box. So this is kind of an ethical question, and this is going to be the debate. So it's 10 minutes each, as I understand, and maybe we can have a follow-up small debate with questions also from the public. So we start with a flip of a coin to see who is going to start. Which side do you choose? Head?
I'll take that. Sure.
Okay, let's see. Me too. Then it's both. Nobody's starting and we go.
It's your turn. Okay, I'll go last. Yeah, I go last.
You want to go last?
Yeah.
You insist? Okay. Fine. It'll actually be me. So the other one. Yeah. It's ready? Yes. Clicker doesn't work. Is it?
It was the farming.
I was okay.
Sometimes it's the farming.
It's not working in the middle of it.
Okay, then we'll just use the computer, then the old-fashioned clicker. Let's just make sure that works. Oh. And that doesn't work. Okay. Sorry.
No, it works. Just a second. Yeah.
There we go, yes.
Sorry?
There we go. Now we're good. All right. Welcome to the talk. Friends, scientists, and science fans, lend me your ears. Are we being maybe a little bit too harsh when we are demanding that medical AI be perfectly explainable? I think if we were to hold HI, that is human intelligence, to the same standard that we hold artificial intelligence, we would find ourselves falling woefully short. Human intelligence is notorious for being unreasonable, irrational, and sometimes downright unexplainable. Even in ancient times, people knew that human reasoning is flawed and biased. Take, for example, the famous story of the fox and the grapes. In this story, a fox sees some grapes, looks at them and thinks, delicious, these grapes are really sweet. So he goes up to the vine, jumps up, and realizes, oh, he's just too short. He can't get those grapes. Being a little bit pissed off by his experience, he walks away and goes, oh, those grapes are sour. No good. So what was it? Were they sour? Were they sweet? The real answer should have nothing to do with the fox's emotional state. Yet we're all like this. We are all affected by our emotions and we make decisions based on them. And it gets even worse. There have been many psychological studies done over the past, let's say, 50 years that have shown that human attention is also limited. This is a scene from a study that was done 20 years ago where the participants were given a simple task. They were supposed to count how many times a basketball was passed between the players with the white shirts. A little bit tricky because some players have black shirts, some have white shirts. So you need to focus. The surprise is that halfway through the video, there's a person in a gorilla suit that shows up, walks into the camera, beats his chest, waves to the audience, and walks off. The shocking thing is, over 50% of the participants didn't see the gorilla. They were so busy counting the basketballs, looking at people, they didn't see that gorilla at all. It's completely obvious. So what does that mean in medicine? What are the gorillas that we have in medicine? Could it be a cancer, a heart disease, a COVID diagnosis that we're missing? Does this happen? Yes, it does, in shockingly high rates. A study in 2014 done in the US showed that there was a misdiagnosis rate of 5%. So in the USA, that's 12 million cases annually of people being misdiagnosed. And that is actually twice the entire population of Norway every year being misdiagnosed in America. So please let us be a little bit kinder to AI and acknowledge our own limitations. Because there are some cases where even completely unexplainable AI, a complete baked black box, you don't know how it works, can be useful. And one of them is very similar to using a dishwasher in your home. When you use your dishwasher, you happily put your dirty dishes into it, because when you run the program, you can look at your dishes and see if they're clean. You don't have to care how the dishwasher works. I don't know how a dishwasher works. I just push the button, I get clean dishes. Good enough. So are there some medical situations that are like this? Yes, there are. For example, in cardiology, there's a very simple yet very often used calculation called the cardiac ejection fraction. It simply counts the volume of blood that is ejected by the heart when it pumps, compared to the volume of blood, total volume of blood in the heart. And this calculation can be used to guide treatments such as pacemakers that are implanted, or drugs, or other things. However, doing this calculation takes a bit of work. You need to go into some medical images of your patient and draw some circles. So where is the blood, and when the heart was squeezed at its most, you need to draw some circles there and compare them to when it's at its biggest. So that takes a bit of work, and guess what? AI can do that. We have lots of these circles, databases of them. If we train an AI to do it, you don't have to care how it works. You can just look at the circles, verify for yourself. Are we getting the blood? Yes, we are. Great. So what could be done with this kind of unexplainable AI? Well, there's more time with patients. So I've done a simple calculation to look at how much time could be saved. There's actually 27,000 cardiologists in Europe. If we just say that they have to do 2 ejection calculation fractions per day and spend 15 minutes doing that, well, that actually adds up to an entire 14,000 hours each day, just doing these simple calculations that could be mostly automated. So what could we do with those 14,000 hours? 14,000 hours? Well, we could do another 3,111 open heart surgeries.
They're not done with the same people.
You get your turn. Okay. Are there some other medical tasks that maybe less trivial? Actually, yes, there are other tasks. So, for example, antibiotics discovery. There was a study done in 2020 in which an AI screened 107 million molecules. I think any biologist who works in the lab would not want to do this manually. That's pretty boring. So the AI was able to just take this mass of potential antibiotics and just pick out the right one. And just like with the dishwasher, there could be a simple test that could verify that this completely unexplainable AI was doing its job. In this case, it was a petri dish where some bacteria were introduced, and the drug was introduced, and lo and behold, the antibiotic had gotten rid of the bacteria. Great job, AI. Who cares about the explanation? It just works like a great dishwasher should. So that was the first case. I think we can all agree that that's very useful, but it goes further. Sometimes AI can produce the best results. And if you're a patient, sometimes the best result is really what you care about. And this is kind of an, let's say, an ongoing process. Are we there yet? Who knows? But if you look at the trends of history, you will see that it's oftentimes the machines that we have. This man here is named John Henry, and if you're American, you may have heard of his legend. He engaged in a competition with a steam-powered drill in the 19th century. So his job was to pound iron spikes into a railroad to build railroad tracks. And when this machine was invented, it was threatening his job. So they arranged a competition. And the legend goes that he actually beat the steam-powered drill, but worked so hard that he died afterwards as a result. Nowadays, nobody is going to try and use a human being for a job like that. We're all using machines. We would clearly go for the machine. And who knows, maybe some years, we will be in that case medically, where no one is going to ask a human being anymore for some important and complicated problems. So do I have an example of something like this? Yes, we have some actually work right here at the University of Oslo on histopathology. So this is a slice of a tumor that was removed from a patient, colorectal cancer tumor. And the big question after you have the tumor removed is, well, is it going to come back? And how long do you expect to live? And do you need to be given more drugs or other treatments? And if you just look at the slide, there's a lot of information there. And human pathologists have tried for many times to analyze this information. And the results have been, I guess, very mixed. But now there's been a group at the UIO that has done this with AI. So just fed all this information into an AI and tried to predict who will survive longer and who will survive shorter. So just taking a figure from their study, and their AI was able to group the patients into 3 groups. So green was a good prognosis, red was a bad prognosis. And so you have the survivor here on the y-axis and years going on the x-axis. And indeed, over time, the patients that had the green prognosis had the best survival, and the red patients had the worst survival. There's no explanation, but if you're in that situation, maybe you want the best answer, because your life depends on it. Finally, there's one more point, which I hope will inspire you as scientists, and that is that we might actually learn something from black box AI. In this case, it's a bit like having a robot scout. The robot goes off into the woods and explores something for you and brings back some interesting information. You may not be able to get an explanation yet, but you can follow it up and learn something interesting. This happened in a study on optimal retinal imaging. So close-up photographs of right into the eye. So close that you can see the blood vessels and other structures in there. So in this study, the scientists wanted to predict the cardiovascular risk just by looking at pictures of the eye using AI. And to do this, they looked at the risk factors that we already use today, like age, gender, smoking, diabetes, BMI. We have good risk calculators that can calculate your risk based on this. So the amazing thing is not that we can predict cardiovascular risk with these factors, we know that already. It's actually the fact that these things are actually detectable in eyes. Before this study, nobody actually knew that men and women had different eyes. That is something, weird. And now that the AI has figured that out, we can look at it and do some more studies and learn something new. So I'll leave it for you to decide then. A black box can actually be a doctor's best friend and our best friend, because human reasoning is flawed, because
Perception of reasoning.
the outputs can oftentimes be easily verified and it's still useful even if we don't understand it. Sometimes AI produces the best results, and sometimes we could learn something which we would have normally ignored. Thank you very much.
hold on that. Now we hear the reply from the cardiologist. Well, we obviously don't need the picture of people's eyes to know if they're female or male or what age they are. We have other ways to determine that. In defense of Gabriel and this debate is kind of constructive because I do use AI. Spoiler alert. Of course, we're all exposed to AI in everyday life, and at this time we are all wowed by all the cool stuff that you can do with AI. For example, image generation, and in a couple of past few weeks we've seen the ChatGPT thing, text generating AI that can answer your questions and generate all kinds of textual output. Super amazing, and why shouldn't this be applied to medicine? And of course we are applying it to medicine. We're using tools that are enabling us to identify fractures, do all kinds of other cool stuff, and imaging is kind of central to what we're doing in a lot of medical AI fields. It started, let's just go back for those who are not familiar with imaging and AI. One central challenge was the ImageNet challenge, which was introduced in 2010. 11 million images which were tagged with their content or classified as containing a leopard or a ship or whatever. A challenge was constructed to make a machine a software that could classify images as well as humans can. And what's really interesting is that in a few years after this challenge was introduced, in 2012, the first neural network was entered into this challenge, and that was a quantum leap in the accuracy of those software systems. And just 3 years later, in 2015, the first AI or machine learning or deep learning tool could classify images better than human. Now, there's a philosophical question that's lurking in that last sentence, because the ground truth, the actual labeling of these images were done by humans. So and the machine is supposed to do what we told it. So can it be better? Like, philosophically, it's interesting. So typical tasks for imaging AI is just, for example, just labeling, finding this is an image of something, a cat or a tumor or whatever, or constraining what part of the image is the cat, which is called localization. Or you can identify several objects in the image, or you can just outline what different types of, like Gabriel actually showed us. Segmentation, finding parts of an image that are of interest. They're really good at it. All those models are really good at doing that stuff. They're like 99% effective now. But when you throw in an object in some kind of situation where the orientation of the object is unusual, this is a data set that's called ObjectNet. You saw the challenge where like 99%, all the AIs were 99% perfect. They failed by 50% when you show them this, because the hammers are not in the place where you expect them to be, or the mittens are not where you expect them to be. And in medicine, you sometimes see unexpected things. And this is the suitable for work version. There are others that we can't repeat. Also, some things are challenging. Sometimes it's very hard to say what's what. This is a meme from some years ago. Can you tell the difference between the puppies and the muffins? Or which is the dog and which is the mop? I'm not sure, actually. This was studied in 2016, I believe. And AI at that time did misclassify a couple. Maybe not that, well, it's not that important when it comes to puppies and muffins, but if it's about a tumor or a medical decision, it has some more consequences. This is a test of some of the high-end networks. When applying ObjectNet, these are all 4 images of Band-Aids, which is a very common medical device. But networks think this is some kind of duck or a paper towel or a slug or a walking stick, actually, and with very high confidence. So there's still some stuff to be learned by these networks. Back to medicine, you probably remember a couple of years back when COVID arrived, and everyone in computer science wanted to, and should be commended for trying to help, but everyone tried to use AI to improve the diagnosis of COVID, for example. And there have been some interesting meta-analysis of all those results, and this study went through 2,000 publications, found about 300 models, tested 60 or 70 of them, and the headline says a lot. The models were not, well, basically were not useful. Obviously, one of the problems with AI, and especially with machine learning, although let's talk about it's deep learning, we're talking about not AI as let's talk about deep learning. You need a lot of data, and when you're training AI for medical purposes, I think we should say that scientific methods still apply. You have to make sure that your data isn't biased, that it's generalizable, etc. And there was a lot of methodological failures in all those models, and none were useful. We do have a little bit of the same problem now with tools that are commercially available, where they're trained on the data set from for example, one hospital or one region, and when you apply it in a different hospital or different region, you don't get the same results because of the patient selection, etc. So medical AI is very complicated. And it's complicated when you have this result, when the AI just says no. What do you do when the computer says no? You can't argue with the AI. It's not good. And sometimes it just says yes. This is a Forbes study of, they tried phones 1 year back, the previous generation of high-end phones, tried the facial recognition, and 5 out of 6 high-end phones let them in with a printed head. I don't think you want that. This is a surgical robot. This system here is these arms are controlling normal surgical tools. Many people think that surgical robots is that self-contained thing, but it's actually a console where the surgeon sits, controlling all those things via remote control. It's not a self-contained thing, but there's a lot of talk about using AI to control robots. I'm not sure how many here drive or have driven a Tesla. Let's see if that works. Let's see if this works now. Well, it doesn't. Of course it doesn't. Because we changed so many times. No, it doesn't. Crap. We posted this. It's not on my computer anymore. This is a video showing the self-driving Tesla, which takes a U-turn or a turn into a cycle lane and drives over some cones. It's supposed to be very intelligent. I wouldn't want that kind of intelligence doing my surgery. Anyway, there's also the potential for hacking or fooling or gaming systems. It's been demonstrated that you can add stuff that looks like noise to a signal that we can't perceive, but you can easily fool an AI into misclassifying stuff. And humans will do things like that to an AI system. There are lots of famous examples where you used, one famous one is in human resources, where people wrote their CVs with white ink on white paper in their PDFs. And when they submitted their PDF for automatic evaluation, they got a very high score because in the white ink on white paper, it said that they were a very qualified engineer. So the AI thought, oh, this is a good candidate. And you should probably be rewarded for being creative. But and this is, we're pretty good at facial recognition. This guy, that's him, but he can print, what's it called? Glasses. Frames of glasses just that and wear those on his nose, and the computer thinks he's Jennifer Lopez. So there are small tweaks you can do to the input that have dramatic effects on the output. And not all of those tweaks are visible, and they're very hard to detect. So I think we need to understand what the medical AI does, because AI tools fail in a very weird, often in weird and unexpected ways. I think we need to trust something before we trust the decision. And I think humans will game or hack systems if they can. Yeah. Thank you. So.
Yeah. Gabriel, do you want to add something to this?
I think we can take questions.
Okay. Let's see.
Yeah.
Yeah.
Yeah, the microphone.
Oh, give him
I can do it. Maybe you can share.
You want to do it?
and then we can
Well,
share.
Share
there's
with
very
Yeah,
few
yeah.
arguments about why we shouldn't use an explainable AI. Mostly that models are not perfect. So, like, what is wrong with a clinical trial? If you have a clinical trial that shows there's 1% mortality with a completely unexplainable model, there's 10% mortality with people, you would say, no, doesn't matter how many people die, models are not perfect.
Well, yeah, you're right. To be honest, we did this kind of like an entertainment thing. So yeah, I'm just saying that medical AI. Yeah, as I said before, scientific methods apply, and we already have a few RCTs where AI has been applied. For example, in imaging, where you compare the results of AI image processing to the human image processing, where the AI, not surprisingly, actually does better. And yeah, where do I stand on this? As long as Gabriel said, when you can see the result and it's when it's reasonable, fits with the knowledge, and you can double check it, it's good, right? No, you don't need explainable per se. You need I think all the scientific communities think about having a human in the loop inside medicine, so you get the output and you get the kind of verification.
Is there time for another question?
Yeah, sure.
Regarding this analogy with the dishwasher, I disagree. I mean, like, even a dishwasher is not just one button. You have, for example, many programs you can choose. And so everyone that uses a dishwasher knows. And I mean, the point is that the user like that use a dishwasher, right? You have these choices. And what program do you choose? So to make that decision, you need to have a certain knowledge of roughly, not maybe specifically, what the program does, but you need to know a little bit. What's the temperature? How long this lasts? How many, for example, if you want to save energy, how many kilowatt hour it uses? All these things. So maybe none of this, the debate of course for purpose of today was black and white, but maybe we need to train a little bit the user in knowing, like, to make the best decision. What's the best algorithm in this case to use? So the same with a dishwasher, you can put your dirty dishes, but let's say if you put something that is, I don't know, there is a part of plastic and you use a program where the temperature goes to 70 degrees, you're gonna get something out that is clean, but it's completely not done. So maybe not so good. But if you, and it seems to me that humans are a bit better than the machines in terms of context and understanding, like, so.
Not just a bit.
More than the machine, right? So we're a bit more, something that is difficult for a machine.
Oh, I think you've answered the question. And yes, machines are much better at context, and I think if the operator uses it in the right context, we can get a good result. And in terms of getting the right buttons and everything, I trust that with these years of medical training that our doctors go through, that they knows something about which buttons to press, and but of course, getting some extra AI training might be very helpful in the future.
Yeah, so we totally agree that context is very difficult for AI at this point. Maybe it's going to be better, I don't know. But is there enough data to actually train every model for everything? We know that AI is very, at this point, with the current techniques we have in machine learning, deep learning is very data hungry. And it's difficult to actually just get the data inside the medical domain to train models that are useful. Medicine is a difficult field. It's a disease is not an entity in itself. A disease is something that a human experiences. And the classification of disease changes over time, and there are cultural differences, there are economic differences. There's lots of different stuff that goes into getting a diagnosis. The criteria for diagnosis changes a lot. I'm a cardiologist. I look at heart disease and myocardial infarction, heart attack. The definition of a heart attack has changed several times in 20 years of life as a doctor. And when you use criteria that were applied to a patient in 2020 or in 2000 to treat a patient in 2010 or 2015, you'll probably get a different answer. So the models need to be always need to be updated, and it's a very big challenge. Also, just getting access to data in the health domain is very, very challenging, as everyone here probably knows. So yeah, but we're both actually optimists. But also, how many here drive a Tesla or have driven a Tesla? Okay, so if you haven't, it's a good cure for AI optimism. Because they have something called autopilot, and that autopilot sees stuff that you don't see, including cars that come out of tunnel walls, etc. So it suddenly does emergency maneuvers away from stuff that isn't there, and suddenly brakes for stuff that isn't there, etc. Which is kind of interesting. Yeah.
Thank you. One more question here?
Yeah, it's a bit of a philosophical question. I'm going back to that graph that you showed for the prognosis of this cancer. So there was this blue curve which you never talked about, the undetermined one. My question is that if we go more and more towards AI-driven medicine, and if that becomes more common, will we fail to train our doctors to then treat those undetermined situations which are not being treated by AI anymore?
That's for you.
Right. So I think what you described sounds actually very nice, very optimistic. If we have an AI that kind of acknowledges its own limitations, and there are some techniques for doing this, then of course, then if we know that the AI can't give us an answer, then definitely let's say you could try a different AI or a doctor or whatever else. What you described sounds actually quite nice. And are we quite there yet? I think it's still a work in progress. Yeah.
Let me try a variant. I was thinking of the training. Well, we're talking about the problem in radiology, which is the first field in medicine where we actually apply a lot of AI, and also trying to do that. And one concern that people have is that the training of doctors would suffer if the junior doctors do not see a lot of the basic stuff anymore. If you just let AIs do all the preliminary work and you just leave the complicated cases to the experts, then will you be able to train enough new experts? That's actually a problem that radiology departments around the world are thinking about at this moment. Because you still need people to double check, and it hasn't been solved. It certainly hasn't. I think a lot of us think about AI and medicine more like autopilots in planes. It's like you let the pilots, they're still there, and they can take a step back and kind of keep a broader overview of what's going on, rather than doing all the things. as I said, at this point, everyone is kind of suggesting using a human in the loop kind of approach where the AI does something and you double check it, but making sure that we can train the next generation of experts is a good thing.
Because there will be rare diseases for which we will never have enough data to train an AI.
Yeah, and obviously with more personalized medicine and precision medicine, all the bins are smaller. We're talking, we used to talk about heart failure, now it's heart failure with preserved ejection fraction, with reduced ejection fraction. We're always making things more and more specific, and that means that we have fewer and fewer individuals in each bin, which makes it harder and harder to train those models and make those. Yeah. So it's kind of a self-limiting thing, I guess. Maybe unless, I don't know, maybe it's going to be it's like a Planck uncertainty at some point.
Thank you.
Okay. One more question? Yeah.
I'd be interested to hear, so like as far as I'm concerned, informed the current kind of status of the regulation on AI in actual clinical use, according to like the FDA, in some things to treat it as medical device, as you say, which is like computer-like assisted decision support. And I would just be interested, are you kind of agreeing with the regulation is heading? So basically, like if you want to put out a new AI device, you have to basically prove that it's like the clinician plus AI is better than the clinician alone, which kind of seems like a sensible thing for me, but what do you think?
Is it a question? I can answer it, but
I guess one thing to think about is that, you know, not everybody in the world has access to a big hospital with highly trained specialists. So there is definitely a potential to carry over some expertise from these specialists in the form of AI to, say, rural or developing areas. And in that case, you wouldn't need to beat the doctor, because for many people there simply is no doctor.
Good point. The regulations at this point that I'm aware of are the EU regulations for the medical device regulation, which became Norwegian or total European law a couple years back. And then the FDA has their own regulation as well. Now, software, in both those regulations, software is now a medical device, just like a pacemaker or a hip prosthesis or something that you implant surgically into a patient. Which means that there are now requirements for, you have to prove that it works as you expect, and you have to, after you have, excuse me, after you've put this device to use, you have to follow up on the use and see that if that still holds, that still does what it's supposed to do when it's been implemented over years. And those are, I think those are good requirements, but there's a problem in that it makes it very much harder for people like us in a small country with small businesses, etc., to actually get through the certification. So this gives large companies and large economies an advantage in the competition actually to make stuff, which I don't really appreciate. But it's good that there is a requirement that software that, as the EU says that if software affects your diagnosis or treatment or prognosis, then it's a medical device. So almost any software that is used professionally in healthcare will be inside the medical device sphere. And AI definitely, when you use it for image processing, etc., definitely a medical device. I think it's proper that we think of those devices that affect your lives as something that we have to have surveillance of and make sure that they work as they're advertised. So, but it's a bigger hurdle for the software industry, definitely.
Thank you very much. I think we have to call it a night. So big applause for.
Our friends again.
So thank you very much, everyone, for showing up, and please follow Pint of Science on social media, and please attend our festival in May if you'd like, of course. So thank you for.
Blod, seriøst, vet du.